Enzyme kinetics. Measuring the Michaelis-Menten kinetic parameters of an enzyme is how scientists characterize and compare enzyme-catalyzed reactions. The overall enzyme reaction can be represented by the following formula, with the enzyme E and substrate S reversibly associating, reacting, and rapidly dissociating into the enzyme and product P. If we graph the concentration of enzyme-substrate complex, and product over time, we would see an initial burst of product formation that corresponds directly to the first step of the reaction, enzyme-substrate complex formation. This is called the pre-steady state period. After the pre-steady state period, the reaction reaches steady state, where all enzyme reaction steps are occurring at a steady rate. This is the steady state period, and during this period, the substrate concentration declines steadily as product is formed. By plotting the change in product concentration during the steady state period and finding the slope of the line, we can calculate the initial velocity of the reaction. Initial velocities are important for finding Michaelis-Menten parameters. When we determine the initial reaction velocities for different starting substrate concentrations, we see two trends. In these experiments, the enzyme concentration and the volume of the reaction are both held constant, whereas the substrate concentration is varied. First, the higher the initial substrate concentration is, the higher the initial velocity. Second, although initial velocities increase, the increase is not as great at higher substrate concentrations than at lower ones, because at high concentration, we are beginning to saturate the binding sites of the enzymes in solution. For this reason, when we plot initial velocity versus initial substrate concentration, we get a hyperbolic curve that is characteristic of first-order Michaelis-Menten kinetics. Using this plot, we can now determine other important Michaelis-Menten kinetic parameters. The velocity that the curve approaches asymptotically is the maximum possible velocity of the reaction, or Vmax. If we divide the Vmax by 2 to find half the maximum velocity, the corresponding initial substrate concentration is known as the Michaelis constant, or Km. Increasing or decreasing the enzyme concentration will change the maximum possible velocity, but will not change the Michaelis constant. It's a constant property of the enzyme reaction. Another way to look at the same data is to plot the reciprocal of the initial velocities and substrate concentration. This converts the data into straight lines and is known as the Lineweaver-Burk plot. Lineweaver-Burk plots make it easier to obtain the Michaelis-Menten parameters. The y-intercept corresponds to the reciprocal of Vmax, the x-intercept to the negative reciprocal of Km, and the slope is the ratio of Km to Vmax. Another way to compare enzymes is to determine the maximum catalytic activity under saturating levels of substrate. This value, called the turnover number, is defined as kcat. The kcat for a reaction describes the catalytic rate, but it does not reveal the catalytic efficiency. In the case of the enzyme fumarase, the Km for fumarate is 5 times lower than it is for malate, yet the kcat values for these two substrates are nearly the same. This results in a specificity constant for fumarate that is about 4 times higher for the enzyme than when malate is the substrate. This higher efficiency means that fumarate is the preferred substrate of the enzyme.